Hello, I'm Maliha from the University of Liverpool, and today I'm presenting our work on aging of human visual system and its effects on contrast vision, especially in context of high dynamic range displays and how age and mean luminance jointly affect our contrast perception. I'll first discuss the effect of aging on contrast sensitivity, that is at threshold level, and then I'll briefly present our data for contrast matching in subthreshold levels for different age groups as well. So I'll begin with describing how we quantified contrast in our study, as there are various definitions of contrast. The most common one, called the Weber contrast, is the difference in the intensity of the stimulus with respect to a background. And there's Michelson contrast, which is commonly used in stimuli with adjoining areas of different intensity, like a sinusoidal grading. Now, these definitions are suitable for our stimuli where only luminance is varied against the background. But in our work, we are using chromatic stimuli as well. So we are using a measure of incremental cone contrast. The individual L, M, and S cone responses are pooled together so as to give a vector length model of three-dimensional contrast. And using this definition of contrast, we collected contrast thresholds for different stimuli to form contrast sensitivity functions, or CSFs. I'll now give you some brief details of the experiment methodology. We use stimuli spanning 3 color directions: achromatic, red, green, and yellow, violet, and 5 different spatial frequencies. And because the stimuli are fixed cycle, the stimulus size becomes smaller as we increase the spatial frequency. We conducted our experiments on a custom-made HDR display, both in Liverpool and Cambridge, which can show very high dynamic range stimuli. We showed these stimuli to our observers in a alternate force choice experiment, and the stimuli were displayed at 6 mean luminance levels ranging from 0.02 to 2000 candela per meter squared at a viewing distance of 91 centimeter. And there were two different groups of observers, young and old. Both consisted of 20 observers each, and the mean ages were 33 and 65 respectively. Now let's look at the results. These are all the CSFs that we collected. The rows are the 3 color directions and the columns are different luminance levels. Each luminance level is 10 times higher than the previous one in candela per meter squared unit. And these icons here, inspired by Stockman and Sharpe 2006, represent example luminances in the corresponding order of magnitude from quarter moon, which corresponds to around 0.02 candela per meter square, to a cloudy daytime sky, which corresponds to 2000 candela per meter squares. And the red curves are the average from the young observers, and the blue curves are from the older observers. And we can see that the older observers, or the blue curves, have lower sensitivity across all conditions. And from this, we can infer that contrast sensitivity decreases with age roughly by 0.3 log units, or 3 decibel. But the cause of this decline is a bit of a controversy in the area of vision research. And the focus of my talk today is to show you what aspects of human visual system aging might translate to this reduction in contrast sensitivity. Now, aging affects various stages of the human visual system, which can be broadly categorized as optical, sensory, and cortical. So optical factors, or pre-retinal factors, might include all the medium that the light passes through before reaching the retina. Sensory factors involve the changes in retina as well as the pathways that convey information to the brain. And the cortical factors involve all the age-related changes that happen in the visual cortex itself. Now, there are two opposing camps in the vision science community. One that believes that the bulk of contrast sensitivity decline with age can be explained by optical or pre-retinal factors, and the other one that attributes changes in contrast sensitivity to neural factors mostly. Before going further, I would like to briefly show what changes happen when the optical elements of the eye undergo aging. Here's a figure from Sarah Lerman's book, Radiant Energy in the Eye, which shows how the lens yellows or browns as we age. And this happens because of the accumulation of certain pigments in the eye, And you can see how around the age of 50, this process begins to accelerate. And this happens even in the absence of any kind of discernible optical disease. Due to this, the total amount of light entering the eye reduces as the accumulated pigment not only absorbs light, but it also causes increase in intraocular scattering of light, which further reduces the amount of light that reaches the retina. Another very important physical change is the construction of pupil, also known as senile meiosis. This figure here demonstrates the changes in pupil size with age. The pupil diameter is plotted against age for two different light levels along with their 95% confidence interval. We know that the pupil size changes to accommodate changes in light levels. It gets bigger in dim light and it gets smaller in bright lights. Now, two trends to notice here. First, pupil size decreases with age. That happens for both bright and dim light. Secondly, the pupil flexibility changes. So for a 20-year-old observer, we can see that the difference in pupil size between bright to dim light is much higher compared to an 8-year-old observer. So not only the pupil size gets smaller with age, the range of sizes that it can go from also changes. And so to summarize so far, here's a very simplistic representation of light entering the eye, passing through the pupil and the lens, and then finally reaching the retina. So when pupil size gets smaller with age, less light enters the eye. The lens also becomes more opaque with age, which further reduces the total amount of light reaching the retina as we age. And so the method so far is retinal illumination decreases with age. Our wheel has quantified this and shown that a 60-year-old eye transmits only 1/3 of the light that a 20-year-old eye can pass. But does that affect contrast sensitivity? To find out, let's go back to the results we saw earlier. I will now compare the CSFs, or contrast sensitivity functions, of older observers with CSFs of younger observers at 10 times lower luminance level. So I'm effectively shifting the whole CSF curve of younger observers towards the right. So something interesting happens in the 20 to 100 candela per meter square range. That is, the CSFs of older observers perfectly match the CSFs of younger observers at 10 times lower light for all three color directions. So the CSFs of older observers at 20 candela per meter square approximately match the CSFs of younger observer at 2 candela per meter square. And the same thing happens at 200 candela per meter square. This shows that reduction in retinal illumination with age due to optical factors can in fact explain some changes in CSFs. But can we model it? So we began by modeling contrast sensitivity function as a function of spatial frequency, mean luminance, chromatic direction, and stimulus size. And we fitted this data for the young observers only, and we got these results. So the model is a pretty good fit. The mathematical details are not the scope of this talk, but it's basically a log parabola modified to include spatial integration with stimulus size as well. So in this very simple model, I plugged in 1/3 the amount of light as predicted by our wheel while keeping everything else constant. So here, instead of 22 candela per meter square, I plugged 6.5 candela per meter square and so on. Here's a table that shows how the retinal illumination decreases for older observers when compared to younger ones. So at 20 candela per meter square, a younger observer might get approximately 20 candela per meter square, but an older observer might see only 6.7 and so on. And so the results we get are here. You can see that for lower luminance levels, the contrast sensitivity of older observers is sufficiently predicted just by reducing the luminance to 1/3. And this is consistent for all three color directions. And the model predictions are well within the standard deviation of the data. For higher luminance levels, however, the lower sensitivity for, especially for higher spatial frequencies, is not explained just by reducing the mean luminance levels. And so to explain the data completely, we had to fit the remaining differences in contrast sensitivity as neural factors, which account for the differences in sensitivity at higher spatial frequencies. Before moving on, I would like to point out that we do not have any empirical data yet to conclusively prove how much of the contrast sensitivity decline is accounted for just by the yellowing of lens and pupil constriction. And we were not able to replicate the findings of RVL, where they have shown a 60-year-old eye only transmits 1/3 the amount of light compared to a younger observer. From our estimate, this reduction in retinal illumination from lens and pupil was just about 10% and not 70%. But the contrast sensitivity data does suggest a high reduction in retinal illumination, the causes of which are not clear at the moment. So far, we have talked about contrast vision at threshold and how age affects our contrast sensitivity. But what happens when we go beyond threshold? Is suprathreshold contrast just as affected by age or not? First, I'll briefly describe what contrast constancy is, which is a term frequently used in context of contrast matching and suprathreshold vision. Let's say we match the physical contrast of pattern 1 of a lower spatial frequency with a higher frequency pattern 2. The apparent contrast will be the difference in physical contrast, C1 here, and the threshold contrast for the specific stimulus. So the left and right side of this equation represents apparent contrast for the two different stimuli. In case of contrast constancy, the two apparent contrasts will be equal in magnitude. Now, a brief description of our methodology. We set up two displays side by side with a divider extending from the middle of the two displays to the middle of the observer's eye. So each eye could only see one display. This configuration is called haploscopic viewing. The display with the reference stimulus was a standard RGB screen, while that with this test stimuli was our HDR display. And our goal was to find out whether there's a contrast constancy across luminance levels. So for our reference display, the stimuli was fixed at 200 candela per meter squared. But for a younger observer group, we also used 20 candela per meter squared as a reference, as we wanted to know whether changing the reference background luminance level makes a difference in contrast matching contours or not. We used 3 different spatial frequencies and 3 color directions at 3 different suprathreshold levels, which we will refer to as low, medium, and high. For each reference stimulus, the corresponding test luminance was displayed at one of these background luminance levels. And the observers were asked to adjust the contrast of the test stimulus to match the reference stimulus. And the observers saw something like this. The right eye could only see the reference stimulus and the left eye could only see the test stimulus. So let's look at the contrast matching results. As stated earlier, there were 3 spatial frequencies and 3 color directions. The solid lines in each plot show the contours of equal perceived contrast. The blue lines for the older observers, while the red and black lines are for younger observers with 2 different reference luminance levels, 200 and 20 candela per meter square. And this whole, the whole plot is for a low reference contrast, and I'll show you another plot for high contrast level, which is just elevated contour. The dashed lines are the contrast threshold curves. The clear trend here is that for all conditions, for low luminance levels, we need higher contrast to match a stimulus at a higher luminance level. This is not surprising because for low luminance, contrast thresholds are elevated as well. But the observation more relevant to today's topic is that the differences in thresholds between young and old observers are not carried over to suprathreshold levels. That is, the contrast perception of older and younger observers for suprathreshold vision is not much different. As you can see, there is a significant overlap of the curves between young and older observers. This result is also supported by another study from May et al. And their work studies contrast matching across spatial frequencies rather than luminance. But you can see here as well that as the contrast is increased beyond threshold level, the young and older observer data overlaps. And so a simple subtractive model like Kulikovsky's contrast constancy model is not enough to explain suprathreshold contrast vision. I will conclude my presentation with a summary of our work. From our CSF data, we have seen that both achromatic and chromatic contrast sensitivities decline with age. The impact of age depends on both the luminance level and spatial scale. Reduction in retinal illumination with age play a significant role in determining changes in contrast sensitivity functions. The RMSE values between young and old observers using a model with no age factor was 0.3 log units, but this value was reduced to 0.14 by simply plugging in a lower luminance value for older observers. And lastly, suprathreshold vision does not show any significant effect of age. Thank you very much for your attention. And I would also like to thank Sophie, Rafal, and Yasna for their valuable contributions to the study, as well as for their constant mentorship. Thank you.